everyone. Welcome back to our discussion of cells. So we're going to talk about the most important molecules in the cell now. That's DNA. And we talked about DNA briefly. It's really the central portion of the entire semester. We're going to be talking about understanding of DNA, how DNA works. So DNA, as you already know, contains the genetic information. And if you're, so this is a double helix structure of DNA. And this DNA has something to do with who you are and how you are in this world. So we're going to discuss all that. And in our cells, in fact, in all cells, DNA is arranged in structures known as chromosomes. The chromosomes of a human are different from chromosomes of other animals or other organisms. In humans, we have 46 chromosomes. And you're going to see that number is closely related to a chimpanzee. They have 44 chromosomes. But different organisms can have different number of chromosomes. If you talk about plants, then they can have hundreds of chromosomes or 200 chromosomes. And this DNA is found in the nucleus as chromosomes in the human body or in eukaryotic cells, organisms. So a little bit about human DNA. So like I said, there are 46 chromosomes. They are actually arranged as 23 pairs, which means that you have like one complete set of DNA that has 23 chromosomes, but we have a duplicate. So that's double in copy, meaning you're getting one complete copy from your mom, one complete copy from your dad. And so you have double of everything. So 23 pairs. That includes somewhere between 20 to 25,000 genes. We don't have a more precise number, but that's how we define genes, I guess. We could just have an estimate. And we'll talk about genes as well. And then our DNA, one copy of DNA is made up of 3.2 billion base pairs. So 3.2 billion base pairs, what does that mean? That means every base pair is basically a bit of information. And you're going to learn about the basis of the DNA, which is going to be A's, G's, C's, and T's. But there are 3.2 billion base pairs. And so that's a big number. That's a lot of information, lots of information that information is enough to encode a human being. Eukaryotic chromosomes, we're going to learn a lot about these in this class. So this is an example of a plant cell, and plant cells are a little bit easier to see. So they're just their chromosomes, they tend to be larger, and it's easier to see where the cells are. And because of the dark staining cell, you can see that here. These stringy things, these are the chromosomes. So in humans, one thing that we can do is that we can look at what's called a karyotype. Remember, karyo refers to the seed. Cells that have a nucleus, they are called eukaryotic. So karyotype is looking at what's in the nucleus of that cell. So what they do is basically they have a cell. They use some type of chemical. They stop the cell at a point in its cycle where you can see all of the chromosomes really well. So then they take the cell, they squish it out basically on a slide, microscopic slide, and then they look at it through the microscope. The reason they squish it out because it's three-dimensional, and a lot of these chromosomes would be in kind of stacked up on top of each other. So squishing it kind of helps spread these chromosomes out. So then they can see them all really well. They take a picture of it, and these chromosomes, they don't line up like they are lined up in this picture. So they take the picture of it and they find each chromosome digitally using maybe a Photoshop, and then they start arranging them. And you can see that on this karyotype, the chromosomes 1 to 23 are arranged, as you can see, pretty quickly by size. So chromosome 1 appears to be the largest, followed by 2, and then 3, and then 4 and 5. And then you can see the smallest one is technically chromosome 21, but to some people 22 appeared smaller. So that's how they have them. There's also a set here that's labeled X and Y. This is a chromosomal sex of a person. The chromosomal sex of a person has if they have a 2X chromosomes, they're considered to be chromosomally female. And we're going to talk about how sometimes the anatomy doesn't always match the chromosomes. So to be most clear and correct and specific, I'm going to use the terms chromosomally female because anatomically, maybe they may not be female and maybe they may be anatomically male, but they're going to be anatomically, if they're anatomically female, that's possible as well. But there are cases when that's not always the case. So it's important to remember that they appear to be chromosomally female means they have 2X chromosomes. And then you'll notice that each one of these chromosomes has 2 copies. So remember, we're gonna say, as we were saying before, you have 2 complete sets of everything. And so now here is something that you might find confusing. So it's about the terminology about chromosomes. So when you have a chromosome in your body, it looks like this, and it's an unreplicated chromosome. It's just a single copy of that chromosome. You would have 46 of those in your cells. So you'd have one that looks very similar to this, but because everything is in pairs, there's a process where it's time to make another cell. We have to replicate all the information, and each of the cells is going to get a complete copy of the chromosome, and then it gets replicated. So you would think if you took one chromosome and replicated it, then you would have 2 chromosomes, right? But that's wrong. So you actually still have 1 chromosome, but there's basically, it has doubled. It has doubled, and its information, but it's still being held together. So as a single unit, so it's still considered 1 chromosome. That, that might be confusing for some of the students. It would make more sense to call it 2 chromosomes, but we still don't call it 2 chromosomes. We still call it 1 chromosome that has been replicated. So this is a replicated chromosome, and this is unreplicated chromosome. And this is still a single chromosome, considered still a single chromosome. And then they're called sister chromatids. So basically like twin sisters. I don't know why they're called sister chromosomes, not brothers, but that's how it is. So we have these sister chromatids that are essentially identical copies of the same chromosome, and they're still being held together in the center. And where they're being held together, that's called a centromere. So that's called a centromere. And this is a dot in the middle where it's being held together. And all of this terminology stuff, you're going to have to know really well as we go through this class. Chromosomes, they're typically identified having a P arm, which is a short arm, and a Q arm, which is a longer arm of the chromosomes. And if they appear to be, even though some of the chromosomes may be appearing like they have the same equal size of those arms, but there might be a tiny, tiny bit difference. So again, the centromere is kind of this constriction of the chromosome where it's constricting, where the sister chromatids are being held together. The very ends of the chromosome have name too. It's called telomeres. These are the ends of the chromosomes. And it's an important structure that's, we'll discuss telomeres again another time, but it has something to do with aging. Here's a microscopic picture of this is electron microscope, picture of some human chromosomes, and you can see the arrow is pointing to this centromere. That's where they're kind of constricted. And you can see the P arm and the Q arm of each of these. And where the constriction is, you notice the chromosome is very tiny. It has a little P arm and a much longer Q arm. Chromosomes will eventually remember, if you remember, we said that a single chromosome replicated, it's still a single chromosome. So we have that here. Then the duplication happens, it's still a single chromosome. It has 2 sister chromatids. Then there'll be a point when these 2 sister chromatids, which are being held together by the centromere here, they are going to separate. Then we call them chromosomes again, so single chromosomes again. So now we get to the column of two chromosomes. They didn't suddenly make more. They were the same as when they were on chromosome, but then now they split apart. So we call them two as soon as they're no longer being held together by the centromere. We call them two again. That's a little weird, but that's how it is. So just learn it and deal with it. So we are at that process where they're being held together, where they go from being held together and then they split apart and they become two separate ones. This is part of the mitosis. This is called mitosis, the process of mitosis. We'll go in detail about it next week. So the DNA is found in the nucleus, and here are some electron microscopic pictures of the nucleus that you can see. So you can't really see much, but these, if you look closely, you can see, maybe still hard to see. Maybe you can squint your eyes and try to see, but these are all the chromosomes. So, and the DNA. So they don't look like, like this here. We'll talk about why they don't look like that. And then we'll talk about mitosis. So I'm gonna stop here and then we'll pick up in the next video and talk a little bit more about cells.